Hi, I'm Mr. Neiman, and today I'm really excited to talk about diffusion and cell size. But first, what is this? Oh, I've got an egg. What can I do with this? So, we talked about eggs last class, and we got to see a demo, but I think I'm going to show it again and talk about it throughout this episode. So let's get started. Alright, so let's talk about what we know about this egg. I know that it came from a chicken. I know that it tastes pretty good. I know that inside it's got a yolk, got this kind of clear part, we got this yellow part to it, and I know that it has a shell on it. So my first question to you is, do you remember what this shell is made of? Calcium carbonate, right? And so calcium carbonate reacts with acids such as vinegar, and it basically reacts by this reaction that you see right here. And we can basically use that reaction then to disintegrate this. So let's see what happens when I put this in some vinegar. But first, I'm going to find the mass and circumference of this egg so that we can compare the egg before the vinegar and after the vinegar. Stay tuned. First, let's find the mass of the egg. So here I have a scale. First thing that I am going to do is turn my scale on, and we should see that it reads all zeros. And then I'm going to put my egg on the center of that scale, and I'm going to take that reading. So this egg has a mass of 53.76 grams. Now that we have the mass of the egg, let's find the circumference. To find the circumference, I'm going to need a piece of string or ribbon and a ruler. All right, so I'm going to take my ribbon, that's all I have, and I'm going to wrap it around the fattest part of this egg that I can find. And I'm going to see where, it's kind of hard for y'all to see this, but I'm going to see where the ribbon meets, and I'm going to mark that spot. Then I'm going to take my ruler, and I'm going to go from the 0 mark on the metric side of my ruler and measure that out. So it appears to be 13.30 centimeters. All right, so we've taken our mass and circumference measurements, and you can see that on the cup that I'm going to use, I have put those down, I've written those down. And now I'm going to pour in some vinegar. So it doesn't really matter how much I have. I just want to make sure that it's going to end up covering my egg. And so I'm going to, I probably should have my egg in there first, but you know, it is what it is. Okay, so now I have my egg, which is covered by the vinegar, in my cup, which is labeled with my starting mass of the egg and the starting circumference of the egg. So I'm gonna let that sit for a while, stew, maybe do an outfit change, I don't know, and then we'll come back and we'll talk a little bit more about what's happening. But in the meantime, let's talk about diffusion. All right, so let's talk about diffusion and then maybe a little bit about cell size as well. So our objectives today are to, 1, be able to explain how and why materials are transported into and out of the cell. We also want to differentiate between passive and active transport and be able to identify hypotonic, isotonic, and hypertonic solutions and which way water is going to flow in terms of the cell and the surrounding solution. So today we're kind of trying to answer, how do molecules with differing properties such as size and polarity travel through the cell membrane? But to start, let's talk about what we looked at before. So we've already looked at what kinds of molecules pass through the phospholipid bilayer of a cell membrane, right? So we talked about the cell membrane, we talked about its structure, we talked about the hydrophilic head, the hydrophobic tail, how they orient themselves. And basically what it comes down to is to move through that one, they have to be small enough to fit. Okay, so to fit through those spaces, they do have to be small. Okay, in addition to being small, non-polar molecules are going to move through the phospholipid bilayer most easily, right? And that's going to come down to those hydrophobic tails. Once a polar molecule tries to go in, it's not going to be able to make its way easily through because it's actually being repelled by the tail. And so it's not able to just like go straight through. And in terms of what do we call this, what I'm kind of going for here is that some things do go through, some things do not. And so we're talking about a cell membrane having a selective permeability. So I wanted to talk really quickly about concentration, right? Because I'm a chemistry teacher and I think that having an understanding of concentration is going to help us out. So when I say words like solution, I mean a liquid mixture that has something dissolved in it, right? So I have a liquid and I've dissolved something in it. The thing that I dissolve is called the solute, and the thing that does the dissolving is the solvent. So typically the solvent is water and the solute is some kind of molecule, ionic compound, something along those lines. When I say concentration, I am talking about how much stuff, solute, is going to be dissolved in a certain amount of solution. So if I make a 1 liter solution, how much is dissolved in that 1 liter solution? Obviously in biology, we're going to be talking about much smaller volumes, but I still want you to think about concentration in terms of how much stuff is in that given volume. So you can see here that I have 2 beakers, right? They don't really look too much like beakers, but you can see that both of them are equal volume, right? So we're comparing 2 solutions of equal volume here because they take up the same amount of space. But what you can see is on the left beaker that we have a greater concentration because we have more stuff or solute dissolved in that beaker than we do on the right side. And I put smiley faces because, you know, I'm a happy kind of guy. So let's get into the meat of this, right? And so we're talking about diffusion, which is the movement of particles of any substance so they can spread out into available space. So kind of one way that I like to think about this is if I were to put a bottle of perfume in the back of my classroom, okay, at the front of the classroom, my students aren't really going to smell it at first. But what's going to happen is there's a high concentration of that perfume back here in the back of my classroom. Those volatile molecules, those molecules that have gone into the vapor phase, are going to start traveling or diffusing to where there's no perfume. So it's going from that high area of concentration of perfume to a lower area of concentration of perfume, which is the front of the classroom, and eventually we smell that, right? So that's what we're going to really be talking about a lot in biology, and really when we get to chemistry as well, is that molecules are going to move from areas of high concentration to areas of low concentration. Okay, and this is called moving down the concentration gradient. So down the concentration gradient. Okay, there is something that I do want to point out with this idea. It almost makes it seem like these molecules have a mission, right? That they are trying to get from high to low. In reality, all molecules, all compounds, all things have a certain movement, right? And we're talking about things that have random movement, right? So there's some randomness to their movement. And yeah, some of them are not going to be headed towards the front of the classroom, but the ones that do keep moving in that direction. So they're really moving in both directions. It's just that the net moving is towards a lower concentration. All right. So I also have this picture. So these 2 pictures here, this one, we see that they spread out or diffuse throughout that solution. Here we see our phospholipid bilayer, and we see that over here we have a very high concentration and a very low concentration or none. It's going to diffuse across that membrane. So this is probably something small and nonpolar. It's going to diffuse across that membrane to areas of low concentration, and it's gonna keep going until they're both here. What I don't love about this picture is it kind of makes it seem like it stops, right? There's still gonna be things that are going to be moving out of the cell and into the cell. They're just really moving in and out of the cell at equal rates, right? And so for every one that goes out, one is coming in. And so that's what we typically are seeing with diffusion. So here, I was gonna draw this out, but I just kind of want you to think about which direction things are going to move. Assuming that those spaces between that barrier is big enough for them to go through, and it is, we should see that the darker colored circles are going to move towards the right. The lighter, or those yellow circles, are going to move to the left. So we're moving down the concentration gradient from areas of high concentration to areas of low concentration. All right? So I'm going to talk about two different types of transport. So when we're talking about diffusion, we're talking about things being transported across a cell membrane. And so we're going to talk about two different types of transport, and that's passive transport and active transport. So when I'm talking about passive transport, I'm talking about molecules going from high concentration to low concentration, just like I said, right? We call that going down the concentration gradient, okay? This takes no energy, okay? This is happening by random motion, right? It's gonna go from high to low because randomly that's what makes sense, okay? But there is no energy that goes into that, which means that we do not have to expend any of our energy source, which is ATP. So the cell doesn't have to use any of that. Okay, and we're going to talk about passive transport a little bit more in a second. Active transport, on the other hand, is moving things across the cell membrane from low concentration to high concentration. Okay, instead of calling that down the concentration gradient, we call that going against the concentration gradient. And super duper important is that doesn't just happen, right? We don't just have that happen. And so we have to put in energy to make that happen. So that means that the cell is going to have to use ATP. Okay, so passive transport, no energy, no ATP needed. Active transport, energy needed, ATP is needed. All right, so I'm going to talk about different types of passive transport. Just a reminder, no energy needed, no ATP needed. So there is simple diffusion, which is what we've talked about already. We've talked about these molecules going from areas of high concentration to low concentration by simply moving down that concentration gradient through the cell membrane, through that phospholipid bilayer. But there's also something called facilitated diffusion. And to have facilitated diffusion, we need a protein. And we call that protein a transport protein. So you might remember that from your web quest that we had, that transport proteins were a type of protein that we saw. Okay, so it allows things that wouldn't just go through easily through simple diffusion. Maybe they're larger molecules or polar molecules or charged molecules, all things that cannot go through that the hydrophobic tail region of that phospholipid bilayer. But they have to have a way through. We have to be able to get them into the cell. We have to be able to get them out of the cell. And so there are those integral proteins that go through that entire bilayer, that phospholipid bilayer, that allow things to move through them from one side to the other. And there are two different types of transport proteins that I need you to know, and that is the channel protein, which you can see right here. It has a little channel, a little place for it to move through, and I kind of call that leaking, right? It's leaking things in or out of the cell. Notice that it is going high to low concentration down that concentration gradient. And we also have carrier proteins, and these go through some kind of shape change. So it might be open to the outside of the cell, when the target molecule comes in, then it's going to cause it to change shape, which would allow that target molecule to then go into the cell. Side note, these proteins look like blobs, right? But they are actually made up of atoms, and they're bonded and they're folded to have the structure that they have. We just kind of showed them as a big blob, okay? But it's all chemistry, it really is, which really it's all fit. All right, so what we see here is we see movement through a carrier protein where we see the shape changing of that protein that's allowing things into the cell. Next to it, we see water actually moving through the cell through a channel protein. So we see it's kind of leaking in and out. Also notice it's trying to keep a balance of the concentration of water on both sides. Okay, so that's actually it for that slide. Okay, so I just wanted you to kind of have some time to think. So water is a very polar molecule, and it's polar because it is a partially negative side on the oxygen and a partially positive side on the hydrogen side of water. Now, it travels in and out of the cell through a channel protein, and I'm telling you that. We actually just saw that in that short little video that I just showed. Now, the question is do you expect the inside of the channel protein to be polar or nonpolar? And then how do you know? Okay, so I want you to think. First, I want you to answer, why does water not easily travel through the phospholipid bilayer through simple diffusion? Okay, now I want you to think about what we learned about transport proteins, what we've learned about attractions between polar and nonpolar, or polar and polar, or nonpolar and nonpolar molecules, and think about what the inside structure of that channel protein has to have for water to move through it. Okay, and I actually want you to write out your response. So pause me, write out your response, and then we'll talk about it. All right, so we would expect the inside of that channel protein to be polar, right? Because we know that it's going to attract those water molecules, it's not going to repel them, it's going to allow them to move through that channel. Okay, and I'm actually going to talk about it at the end of this video some more because I think that water moving through aquaporins is just fascinating. Anyway, so when molecules are moving down the concentration gradient, are all molecules moving in the same direction? Pause me, think about it, write about it, and come back. Okay, they really aren't, right? So we're going to have some that are moving in both directions. It's just that net, so overall, they are moving in generally one direction. But that doesn't mean that we don't have some going the other way. It just means we have more going down the gradient. So next class, you're actually going to talk about osmosis. And so I'm going to give you a little bit of a starting point to some things to kind of think about to get those questions going that you might have for the next presenter. And so osmosis deals with the diffusion of water, okay, across a selectively permeable membrane. So some membrane, or if a membrane is selectively permeable, like the cell membrane is, then it's going to let some things in and not let other things in, right? All right, so let's talk about three different types of solutions: hypertonic, isotonic, and hypotonic, okay? And I'm not really going to go into the reasons behind these at all because that should happen next class. But I am going to talk about what basically happens in each solution. So if I put a cell into a hypertonic solution, then water is going to leave the cell and go into the solution, okay? Basically, you can kind of think of it as there's more water in the cell, there's a higher concentration of water in the cell than outside of the cell. So the water is going to move out of the cell. In an isotonic solution, if I put a cell into an isotonic solution, iso means equal or same, and so the net movement of water is going to be 0. Okay, so cell kind of stays the same. Hypertonic, it kind of shrivels up because the water leaves, right? Then in a hypotonic solution, we're putting the cell into a solution that is less concentrated than what's in the cell. Okay? That means that the concentration of water in this solution is higher than the concentration of water in the cell. So water is going to move into the cell, which causes it to get bigger. Okay? I do want you to think about how this relates to our egg, okay? Because I am going to talk about that in just a little bit. All right. Now, active transport. So when I'm talking about active transport, remember, energy is required. And where is that energy coming from? That energy is coming from ATP, because that's where it's been stored. This is going to be where we go against the concentration gradient. So we're going from low concentration to high concentration. And these require protein pumps. Okay, so I'm not going to talk in depth about this, but just realize that here it's showing that the sodium ion concentration is high outside of the cell, and then inside the cell it's low. But notice how we are pumping sodium out of the cell. Okay, notice that we had to use ATP. So we had new bonds that were broken, new bonds that ended up getting formed. Overall, that's going to provide the energy for this to occur. Okay, so just notice that new bonds were formed here. Okay, you also see that the same thing happens with the potassium, where it's going against the concentration gradient. Here, I want you to identify the processes occurring in each picture. I want you to say what process is moving the material and whether it is passive or active transport. After you have paused me and completed that, then we will go over the answers. All right, so in the picture on the left, we see that simple diffusion is occurring. We're going from high concentration to low concentration across the cell membrane. Okay, that is simple diffusion, which is a form of passive transport. Okay, for the picture on the right, okay, I see ATP. I know it's going to be active transport. And in terms of what process is moving the material, we obviously are doing that through a protein pump. Okay, so we're going to have to pump them through using ATP. All right, so cell size. This is really the last thing. I have a little bonus information at the end here. But cell size, as things get bigger, right, their volume is going to increase at a greater rate than their surface area. So the amount of space it takes up is going to increase at a greater rate than the surface area, so the area where things can actually move in and out of the cell. So what that does is it lowers the surface area to volume ratio. That's not good, because we need things to move into and out of our cell, and the bigger the cell is, the harder that is to happen, because the surface area is not great enough to support it. Okay, so what that means for us is that we need our cells to have a high surface area and low volume, right? So we need a high surface area to volume ratio. So we really kind of like smaller cells, right? Smaller cells that can fit everything in there, allow us to get everything in that we need, and also kind of get rid of anything that we need to get out of the cell. All right, so this is my if time permits little section, because I'm a chemistry guy and I think that biochemistry is fascinating, and so I wanted to talk about a couple of things here. Okay, and that's aquaporins. And so we've talked about water going across the cell. Okay, so here I actually have a video of a cell membrane, right? And we can see that. We can see the hydrophilic heads right here. So those are lined up right here, those hydrophilic heads. And in the middle, we have our hydrophobic tails. Okay, out here we have a lot of water. Now what's going to happen is one of those waters is going to be tagged as a much larger molecule than water actually is, but it's going to show kind of that process of it moving from out of the cell to into the cell. And no, we're not going to watch 1 minute and 10 seconds of it. We don't need that. Come on, I just want to be done. Maybe I'll just shoot another video and post that in there. And just splice it in, because this is boo-boo. All right, so what we can see here is that the water is moving into that hydrophobic area. Now that area is full of non-polar fatty acid, right? And so, so that area is full of non-polar lipids. And so we see that it's really having a hard time, right? It's kind of getting bounced around because it's getting repelled, and it's not moving through at a fast enough rate that we need water to move through. So we have discovered aquaporins, and these were actually discovered not really that long ago in our scientific history, right? And actually a Nobel Prize was awarded for it in 2003, and so in chemistry. And so what you see right here is kind of this structure, because I wanted to make sure that you realize these are not actual blobs, right? There are atoms here that are bonded, and then they have these different structures and folding and interactions that they have with each other, okay? And you can kind of see this top view here where water would move through that channel. Side note that I did want to point out, just because I do think it's fascinating, is like, how do we know that through these channels only water goes through? Okay, and you're like, well, wouldn't other things just be able to also go through? But here is something that is actually really cool. So if you look at this channel here, right here on this protein structure, we have these positive charges. Okay, so what ends up happening is if these ions start to try to go through, or different ions start to try to go through, like sodium, which we need sodium to stay on one end, okay, because we have a pump that we need to do that. We need to have protons and other positively charged things on the other side. We can't let those ions go through. Well, what we end up having happen is if they try to come in here, they might get in, but once they get here, they're gonna be repelled, because positive charges repel positive charges, and boom, that is fascinating to me. Okay, and then I'm just gonna show kind of lastly, really quickly, hopefully, that I'm just going to show this water channel, which is a channel protein and an aquaporin, and we can kind of just see that they are moving in to have equal concentrations of water on both sides of that cell membrane. All right, cool. So for next class, I want you to think about how we can predict water flow through osmosis. And so you really want to think about hypotonic, hypertonic, and isotonic solutions, and which way water is going to flow and why. So now we're going to go back into the lab, and we are going to talk more about your eggs. Hi, welcome back. It's time to take the egg out of our cup and take some measurements. All right, so you saw the egg in the cup. I have since taken it out. I gave it a little wash. I have found the mass, and I have found the circumference of this egg. And so before I do the next part of this lab, I wanted to show it to you, right? So I'm going to come to you, and here it is, right? This is with no shell, right? So the shell is gone. That calcium carbonate has reacted away with that vinegar, with the acetic acid in the vinegar. And now I'm just left with the egg, with the cell membrane surrounding it. And I want to know, what are your thoughts on the data that I have? So go ahead and jot down your thoughts on that data. Why did the mass do what it did? I don't know. Let's think about it. Okay, so in a second, I'm going to go and talk to y'all some more about exactly what happened. But in the meantime, we need to do something with this egg. So what I'm going to do is I am going to take this egg and I am going to put it back into my cup. right? So I have already recorded my data, and I'm going to put it in my cup filled with Karo syrup. I'm going to gently put it in, make sure it submerges. Okay. A clean lab coat is make sure you ain't doing work. Okay, so I now have that in my solution, so I'm going to let that stew for a while. All right, so I'm going to let that go for a while and see what happens. All right, so let's talk about what we saw when we took our egg out of the vinegar. So if we compare our masses from before the vinegar to after the vinegar, we saw that it went up. Same thing with my circumference. But I bet that many of you hypothesized that the mass and the circumference would actually go down. And you probably thought that because we took away the shell. The shell was there, now it's gone, so the mass should have went down. But that's not what we saw. So we have to be able to explain why that hypothesis was not supported and what we really think maybe happened here. All right, so we removed the shell. Then we now had this cell with its cell membrane, right, the membrane of that egg, in a solution of vinegar. I'm wagering here that that solution of vinegar has a higher concentration of water than was inside of the egg. If that's the case, water was able to selectively go from the solution into the cell, which caused the mass to go up, right? And we call that putting it into a hypotonic solution. All right, so I've taken our egg out of the syrup, and I've taken the mass and the circumference. So maybe you should kind of hypothesize about what happened here. Did the mass go up and down or down? Did the circumference go up or down? And then I'm gonna kind of show it to you, right? So let me get down here again. And what I see here is this is super squishy. It's definitely smaller. It has less mass and has less circumference as well. You can just tell like how squishy it is, kind of cool to hold in your hands, right? And so what I need to do is do one more step. And so I'm gonna come back up here for you guys, and I am going to add my egg into some distilled water. So I have added that to the distilled water, and I'm gonna let that sit and stew for just a little bit. Okay, so let's talk about what happened to the egg in the syrup. First, I want you to ask yourself if the hypothesis that you had was supported by the evidence that we have. And so right here I have my evidence for my mass, and right here I have my evidence for my circumference through all the stages so far. And so let's see if what you have kind of matches up with what you thought would happen. So let's also talk about why this happened. So we know that water must have left the egg, right? Because the mass of the egg went down. So what that means is that the concentration of water outside of the cell must have been lower than inside of that egg cell. Okay, so it went from high concentration of water to low concentration of water. And that makes sense because the syrup is very, very sugary. And so it has a high sugar concentration, but an overall lower concentration of water than what I had in my cell. So we saw that the water went out, therefore making the egg smaller. All right, so therefore we would say that the sugar solution or that syrup solution was a hypertonic solution since water went from the cell into the solution. All right, so the egg is out of the water. We have finished our last trial, so let's wrap this thing up. So, like I said, egg is out of the water, and what was your prediction? What was going to happen to the mass and circumference? Guess what? It went up. So let's talk about the reason that it would have gone up. So it must go up because since I put it in distilled water, we know the concentration of water is going to be much higher outside of the egg than inside of the egg. So since the water is going to flow from high concentration to low concentration. The water flowed into the egg, therefore increasing the mass and increasing the circumference. And so you can see it's still pretty squishy. I honestly probably could leave it in there for longer, but I kind of got a little bit impatient. And so that is the experiment. I do want you to consider this experiment when we are talking about osmosis, and think about how it relates to words like hypotonic and hypertonic and isotonic. All right, bye everybody.